I expect you're all feeling really good right now. I will warn you two things. One is that this room tends to heat up. And the other is that you probably won't feel as good when I get done for a variety of reasons. So, you've probably seen this phrase, better living through chemistry. And in fact, most of us wouldn't be here today if it weren't for the tremendous advances we've made in chemistry in the last 100 years, particularly in the field of medicine, but in computers and in just agriculture and almost anything you can think of. Chemistry has been at the bottom of all of the advances that we've been able to make. So we almost can look at, and probably in the future, we might even see this title given to the 20th century as being the chemical century. Certainly a chemical revolution that took place during this time. Interestingly enough, we've added something like 80 to 100,000 new man-made chemicals to the environment. These are chemicals that were never there before. And we've increased the levels of many of the natural chemicals to levels that biological systems have never before seen, which is one of the problems we're looking at. This chemical mixture that's out there has really never been encountered by plants and animals and microorganisms. It's new for them in an evolutionary sense over time. So today we pretty much find ourselves living in a chemical sea. We're surrounded by these chemicals. They're in the air, they're in the water, they're in our soil, they're in our food, and they're accumulating in our bodies. And we have some concerns about that. And I'm going to address some of those concerns today. Now I'm going to break things up into 4 parts. First of all, I want to explain how the endocrine system works so that you have a little bit different picture maybe of what's really going on inside your bodies than you might have had before you got here. And it's important to understand why we as endocrinologists are concerned about these chemicals in the environment. Secondly, I want to look at what we call the stealth pollution now, which is disrupting endocrine systems in our bodies and its effects on water and wildlife. Then we'll extend that to humans. And finally, I want to look at what's being done about stealth pollution and maybe what we can do about it. As I go along, I've broken it up into smaller parts within these 4 areas. And because of the size of the group, I'd like you to hold your questions until I reach the point where I ask you to dump them out. And in fact, you may find that I've planned it such that your question got answered before we got there, but I'll let you know when those opportunities arise for questions. So first of all, I want to look then at chemical communication, the whole process of what's going on in organisms, because it sets a background. Now there's a fair amount of detail that I'm going to talk about, and you don't have to worry too much about the terminology and so on. What I'm trying to do is to give you an image of what biological chemical regulation is like. And then we can go and see how we can mess it up. Now, chemical communication is as old as life itself. It started out in the first cells. It's common to all animals. The same mechanisms we find operating regardless of what level of animal we look at. We see the same things going on in their cells, pretty much. So if I use this cartoon here to represent, let's say, 3 cells, and each one of them is surrounded by a membrane that's composed of phospholipids and with proteins embedded in it and so on, and it serves as a filter or a barrier to control the movement of things in and out of cells, for one thing. And early on, cells had to learn to recognize other chemicals in their environment, particularly toxins, nutrients, and other molecules that are out there. And they do this, through what we call protein receptors that are located on the cell membrane, and other kinds of proteins as well. And that's sort of illustrated in this cartoon. And we also find that cells actually secrete chemicals, and some of those chemicals are what I call bioregulators. They regulate the activity of other cells, and those are again detected through specific receptor molecules that are located on those cells, and consequently affect what other cells do, and that's part of the communication that goes on between cells in our body. And in response, those cells may accumulate some of these toxins and nutrients and things from the environment as well. Now, basically in animals like us, the nervous system and the endocrine system have really specialized in what we call bioregulation. That's what controls everything that goes on in our bodies, pretty much. And turns out there are several kinds of bioregulators that operate in our body. Details aren't real important, but you're familiar with neurons. They actually connect directly to other neurons or muscles or gland cells in the body, and they release chemicals at those sites called neurotransmitters, and those actually then regulate what those organs and the cells in them do. A lot of cells release their regulators into the blood vascular system, and by definition we call those hormones, and they travel long distances through the body to their target cells and produce effects there. And then there are even cells within tissues release chemicals that affect other cells within those tissues. So we have all this chemical communication going on within the body. Whoops. And I threw this table in, which I just passed very quickly. And it doesn't want to back up. There we go. Not for you to really pay attention to it. If you're familiar with the kinds of regulators that we have, proteins, peptides, some of them are modified amino acids. They're all water soluble, which is what this indicates here. Then there are steroids and thyroid hormones and a number of small lipids. They're not water soluble. And it's really the small molecules that we're more concerned about, because these larger ones, particularly all the ones, the top three, they don't last very long in the environment. They don't even last very long in our body. They're rapidly degraded. But these chemicals down here last quite a bit longer. And those are the ones that we first started focusing on in terms of chemicals in the environment that were actually affecting our biology. So, very briefly, how does our endocrine system work as a regulatory system? And first of all, be aware that every aspect of your physiology and behavior is either directly controlled by hormones or it's modified by hormones. There is nothing you could think of that I couldn't tell you that there's a hormone that affects it, because they affect everything. Furthermore, they interact with each other. They don't work in a vacuum. What one hormone does may affect how another hormone works. So they're all related to one another. And they work at very, very, very low concentrations. We're talking about very, very small amounts, and I'll say something about that in more detail later. The way in which a hormone works is that it binds to these receptor molecules that I mentioned before that are found on target cells. And this cartoon represents a target cell, and I've put a receptor on the cell membrane. Sometimes the receptors, it turns out, are inside the cell and not on the surface of the cell. But they work the same way regardless of their location. Along comes a hormone in the blood, and it binds to that receptor, and it sets into play a whole series of biochemical events inside that cell. Fairly complicated kinds of things. And if it turns out that there is enough hormone in circulation, and there's enough of these target cells that have enough of these receptors on their surface, then we get enough of those cells responding that we actually see an effect at the level of the organism, and that we've learned to specifically associate with that particular hormone. Now, it's a little more complicated inside the cell than what I just told you. Okay? It's incredible. These represent a couple of receptors on the cell surface. They interact with a whole kinds of a whole host of chemicals inside, special pathways. They even get down into the nucleus of the cell and affect, by turning on certain genes and turning off other genes. They affect enzymes, they affect everything that's going on inside those cells. So there's a real complex inside that little black box we call the cell that these hormones are influencing. Whoops. And I threw out this cartoon just to indicate that every hormone has a life history, just like an animal has a life history. So that it's born by, for example, being secreted by an endocrine cell. That cell is often controlled by either neural stimuli coming to it or by other things in the blood that are telling it what to do. They could be other hormones, they could be chemicals like calcium or glucose. And in response to those chemicals, the endocrine cell secretes a hormone into the blood. This red blob represents the hormone. And then a lot of things can happen to it. It interacts with proteins that are in the blood, we call binding proteins. This allows a small fraction of it to be free and a large amount to be kept in the blood as a reservoir for instant use by the body. It actually may enter the kidney and get excreted, or it might get metabolized in the kidney to the blue dot and get excreted. It may end up in the liver and get metabolized and excreted, or it may actually get metabolized and changed to another bioregulator that goes back out into the circulation and binds to a receptor on a target cell. And then this line represents just going to its target cell and binding to a receptor and causing some specific effect. So the hormone is born, it lives, and it dies through metabolism and excretion for all practical purposes. We'll show you that diagram again a little bit later. Now, most of what goes on with our endocrine system actually involves both the nervous system and the endocrine system working together, because it turns out that virtually every cell in the body makes hormones, and the brain is no exception. The brain secretes hormones into the blood just like your thyroid gland does. Different hormones, but the same kind of process goes on. And the neuroendocrine system is what controls most of our biology. There are other endocrine mechanisms that are not within this, what we call the neuroendocrine system, but they're important too. So, just to give you a quick overview of what that system looks like, it involves the brain, a particular part of the brain called the hypothalamus that makes these hormones that controls your pituitary gland, which is located right behind your nose. In fact, we used to think it got its name, pituita, from the Greek word for slime, because they thought it made one of the four humors of the body, phlegm. And so they thought that's what its function was. Turns out it's not true. It's actually a really important endocrine gland, sometimes called the master endocrine gland, because it secretes hormones that control our four major endocrine tissues in our bodies: the thyroid, the liver, the adrenal, and the gonads, which is a quick way of referring to ovaries and testes generically. In turn, those organs all produce specific hormones. And in case of the gonads, we're also making eggs and sperm under the direction of this whole system. And that then affects pretty much all the important things that we do. We develop, we grow, we get stressed, we reproduce, and of course we have metabolism going on all the time as well. And these hormones all influence those processes in various ways. Whoops. Now, most of what I'm going to talk about is just going to be this pathway here. But everything I say about this pathway is true for those other pathways as well. We just don't have time to look at all of them. So we're just going to focus on that one. Before we do that, I want to make one more thing, and that is I'd like to compare endocrinology, toxicology, and pharmacology. And then I'm going to open it up to some questions. In endocrinology, we're interested in what we call the dose-response relationship. In other words, what kind of a response do you get as you increase the dose of a hormone? And you can graph these responses, for example, as here. As we increase the dose along here, we get a greater and greater response until finally we get the maximum response that the body can make. It can't do any more, and and so it plateaus out at a certain point. Sometimes, however, instead of just plateauing out, what we find is that the response starts to decrease. This might be due to toxicity. High levels of the hormone may start killing off cells, may start poisoning processes within the cell, so that too much of a good thing, like Socrates always tried to tell us, is too much of a good thing. Also, there's another process that goes on called feedback. And in that scheme that I showed you earlier, the life history, one of the actions of hormones circulating in the blood is to bind to receptors on the cells that control the whole system. It might be in the brain, they might actually be the hormone secreting cell like in the pituitary or in the thyroid or something like that. And this hormone then tells them that, hey, there's plenty of this hormone out here in the circulation, stop making it. Similarly, if the hormone level drops too low, that tells the system to start making more. And that's what we call feedback in an endocrine system. And it's very, very important. It's what tells the body everything's okay. I mean, how does the brain measure growth, for example? How does it know that your femur is getting longer? It doesn't. Okay, it relies on the levels of the hormones that are supposed to cause that to happen. It doesn't know whether you're making sperm or eggs. It pays attention to the hormones that are controlling that process. It measures those and it says, if those are right, then everything else is right. If they're too low, we're going to make more. If they're too high, we're going to make less. That's feedback. It's really, really, really important in endocrine systems. It's the one concept our students get all confused with right off at the very beginning. Okay. Now, in toxicology and pharmacology, toxicology deals primarily with toxic substances and their effects on animals or plants or microorganisms. Generally speaking, they're looking for 100% response. In toxicology, of course, that response is usually the death of the organism or the induction of disease. That disease either being cancer, usually, but it could be a different disease as well. So they're usually looking for toxicity, you know, what dose kills the animal, and then they look down and say, what dose doesn't have any effect? They call it the no effect level. And then what they usually do is say, okay, if that produced no effect in our test, if we say 1,000th of that concentration is probably safe to expose people to. So then we try it on people at that point to make sure that it doesn't do that. So they usually dumb it down by what we call an order of magnitude, 1,000-fold. So if a gram would kill you, we'll say a milligram would be safe. In pharmacology, what we're looking for is the clinical benefit. So our 100% level is that we make you well. Although at the same time, in pharmacology, we're concerned about possible toxicity. That's the fast part of the commercials on television. When they go through this long list of side effects, that's the toxicity that's there, even at the level that maybe is producing the desired effect that we want. Now, the toxicologist or the pharmacologist, as far as that's concerned, for them, dose is everything. Okay, they don't pay attention to anything except the dose. Toxicology, of course, deals with environmental safety of chemicals, and they're regulated primarily today, in the United States at least, through the Environmental Protection Agency, or our good friends, the EPA, as we call them. Pharmacology, the whole purpose of pharmacology is to develop simple, stable drugs to ward off infection, develop simple, stable drugs that mimic natural bioregulators, develop simple, stable drugs that can block the actions of natural bioregulators when you're making too much of something, for example, and to do this with minimal side effects. And of course, they're regulated by the Food and Drug Administration, or what we call the FDA. Now, in aquatic toxicology, which is where most of my work gets involved with, we're talking about the amounts in water of these chemicals. And so we're often talking about, we take into account the fact that a liter of water weighs about 1,000 grams by definition, if not actually. Depends on, of course, what altitude you're measuring, you're weighing your water. And a gram per liter, for example, would be a part per thousand. A milligram in a liter, which is a thousandth of a gram, is a part per million. And a microgram per liter. Did I cover up my microphone? Sorry about that. A microgram per liter is a part per billion. And a nanogram per liter is what we call a part per trillion. Now notice PPT would come out twice on here, but nobody pays attention to a part per thousand. So whenever we say in aquatic situations PPT, we always mean parts per trillion, never parts per thousand. Now, in aquatic toxicology, the focus is really on grams per liter and milligrams per liter, because that's where the toxicities traditionally lie. Okay? And these are considered trace amounts. When they say trace amounts, they're talking about micrograms per liter or nanograms per liter. They don't classically find these producing disease or death. Whoops. Now, in environmental endocrinology, which is the sub-area of endocrinology that I'm involved with, where we're interested in how the environment works through the nervous system and the endocrine system to control development and reproduction and the aging process and so on. And we've worked in all those areas. For us, the important levels, the concentrations we're interested in, are the micrograms per liter and the nanograms per liter, because that's the level at which hormones work in the body. Those are the real levels that we're talking about when we're saying very, very, very low concentrations that hormones work at these conditions, okay? And those other things, we don't pay too much attention to them because they're killing off organisms. Those are toxic as far as we're concerned when we're dealing with hormones. You don't ever want to get to those kinds of doses. That's not real physiology. They're okay for clinical purposes, and lots of times that milligram per liter, or generally pharmaceuticals are administered in milligram level doses because we have to get an effect. And of course the body sees these as strange chemicals and tries to excrete as many of them as possible, as fast as possible. So 90% of the dose that you take within 24 hours is in your urine, not in your body. So that's why we have to give you a big dose in order to get an effect. And hence we get some of the side effects as a consequence. But we're talking about parts per billion, parts per trillion, you know. Well, what the heck does that mean, really? What is a part per trillion? You know, I'm not even sure what a million is, to visualize it. So to help you visualize this, and I'll give you one example. Let's take a pinch of salt. You can all sort of identify with a pinch of salt. And then we're going to drop it into an Olympic swimming pool. And that's about 1 part per trillion of sodium chloride. Really low concentrations. But that's the levels at which biological cells respond to environmental chemicals. Really small levels. And kinds of responses that the toxicologists never even thought to look for, because that's not what they were charged with. So, to endocrinologists, the dose is really important, but as you'll see, there's something even more critical that is ignored by pharmacologists and by toxicologists, at least up until very recently as they've been listening to us. And that is, timing is everything. It's not so important what the dose is, it's when the animal is exposed to it, you can get very, very different responses. And so the endocrinologists and physiologists in general, when we talk about the dose-response relationship, we also have to include time. We have to know when the animal was exposed, because sometimes the exposure can occur decades before we see the response. And the last thing I want to leave you with this section is just this little statement. The fish endocrine system works just like the human endocrine system. So anything we know about the fish endocrine system tells us what the human endocrine system is working, or how it's working. And anything that we know about the human endocrine system tells us what the fish endocrine system works like. That's also true of amphibians and reptiles and birds. They all have basically produce the same hormones, the same systems are there. The same garbage inside that cell is there in all of these groups of animals. We all respond pretty much the same way. Okay, let's move on a little bit and want to do a little history first about Rachel Carson. It was Rachel Carson that got me interested in marine biology, actually, with these two books she put out. But her most famous book probably is Silent Spring, which she published 2 years before her death. She died of complications associated with breast cancer. And her focus was on one particular chemical in the environment, DDT, which at that time was reducing bird reproduction levels. Their egg shells were thin, the young weren't surviving, and we were seeing tremendous changes around the country, particularly in the Great Lakes region, all of which could be tied into the extensive spraying. Here you can see them spraying a beach. Those are people with DDT. And in fact, it was because of her efforts that fairly soon, because of public outcries and all kinds of demonstrations, Congress established the Environmental Protection Agency. Richard Nixon signed it into law. He thought it would disappear, didn't think it was going to take off. He put William Ruckelshaw in charge of it, and he surprised everybody because he decided he was going to enforce the Clean Air Act and the Clean Water Act. They didn't expect he was going to do that, and he made himself really unpopular with industry for a while. But the EPA actually did create an incredible improvement around the country. The air in the cities improved, the Great Lakes improved, the Cuyahoga River didn't catch on fire anymore. All kinds of very obvious visual changes. And we said, hooray! The government now is looking after that. We as biologists don't have to pay attention to this problem anymore of pollution. And I just gave you that quote from Ruckelshaw that said, air and water pollution, solid waste disposal, radiation, and pesticides is what we are all about. And particularly they focused on air and water pollution. They haven't done too much with the other. And so we're all happy the earth is saved. Okay. Now, the EPA has done a pretty good job of keeping the levels of toxic chemicals down. But it's interesting, how many toxic chemicals do you think that the EPA has banned since its establishment in 1970? Anybody care to make a guess? Seven. And one of those got unbanned. That was asbestos, because we couldn't do without it. Your cars wouldn't stop. They haven't found a substitute for the asbestos in brakes yet. And so it's really six. And DDT was the first one. Now, between 1985 and 1995, then, we started getting some disturbing information showing up. Lou Gillette down in Florida looked at feminized alligators that had been exposed to DDT and its metabolite DDE at Lake Apopka in Florida. Now, DDT is one of these substances and its metabolites that stays in the environment for years and years and years. And so it's, even though it was banned in 1972 or 70, right around in there, it's still out there. It's still in our soil. It's still in our bodies. It's still in our food. Theo Colburn, looking at data on the effects of PCBs accumulating in fishes that were eaten by pregnant women, were having effects that they could measure in the intelligence of their children by the time they got to late elementary school, middle schools. Here in Colorado, we were looking at things like DDT and dioxins and insecticide methoprene and its effects on amphibian reproduction and development. And we were also looking at the effects of cadmium from mining operations in all of our rivers here in Colorado and its effects on adrenal function in rainbow trout. And about this time also then, in the United Kingdom, they started reporting feminized fishes downstream of wastewater treatment plants in some of the major rivers in the United Kingdom. And we thought, that's kind of interesting. Wonder if that's happening here. Now, Theo Colburn got a group of scientists together in 1993, and they came up with this term for these chemicals in the environment that were having effects. And they called them endocrine disrupting chemicals because they were interfering with how our endocrine system was functioning. Theo then published a book. I have a copy down here if you'd like to look at it during the break, called Our Stolen Future. And if you've never read this book, you should read it right after you finish reading Rachel Carson's book on Silent Spring, if you haven't read that one. It's a wonderful book, especially the opening paragraph. The term endocrine disruption or endocrine disrupting chemical has been broadened a little bit. Now people are starting to call them emerging contaminants. Emerging contaminants then would include our endocrine disrupting chemicals, but they might be neuroactive chemicals that are affecting other aspects of the nervous system that aren't tied in with necessarily our neuroendocrine system, or they could be affecting other organs in the body. Again, we said there were a lot of these chemicals out there. Most of them have been dumped into the environment in the last 70 years. And there's been a steady increase in the amounts of these chemicals. And we can find them at concentrations of parts per billion commonly and parts per trillion commonly. But only in the last 10 years or so because we couldn't measure them. Our chemistry wasn't good enough to measure chemicals at these concentrations, so we ignored them. We didn't even realize they were there. But biological systems knew they were there, and biological systems can respond to them even when we couldn't measure them chemically. Oh, another test question. How many pounds of toxic chemical waste did the U.S. release into the environment last year? Well, oh, excuse me, this is 2012, isn't it? We don't have the data for 2011 yet. They're still totaling it up. For 2010, any guesses? These are toxic chemicals, known toxic chemicals that we dumped into our environment in the past year, in the year 2010. 10 to the 9th. Yeah, 10 to the 9th pounds. That's a lot of pounds. That's a 9% increase in what we dumped in the water over the previous year, 28% on land over the previous year. Hey, but we're doing better on our air pollution. That was a decrease in the amount of stuff we threw into the atmosphere which currently is where EPA is putting a lot of its new regulations, has been associated with the atmosphere. So we can define then endocrine disruption in the following ways. Some of these chemicals are endocrine disrupting chemicals. We've been able to show that in the laboratory and in the field. And what they do is they mimic or they block how hormones work. So they can either make them better or make them less effective. Typically, they are all small molecules that are very, very persistent in the environment, much more persistent even than the natural hormones. So they might last years, decades in the environment with little or no degradation. Some of these chemicals we've followed them for 40 years so far, and they haven't degraded at all. So we don't know what the degradation rate is in the environment for some of these chemicals. Actually, for most of these chemicals, we don't know anything about them. Some of the ones that we know are endocrine disrupting are estrogenic, some are anti-estrogenic, some are androgenic, some are anti-androgenic, some are anti-thyroid, some are anti-adrenal, some are obesogenic. The obesogen, that's a new term we came up with just a few years ago. Now you don't have to blame it on your inactivity. You can blame it on the chemicals in your environment, that's what's making you overweight. Probably not. But they can add to the problem. Where do they come from? They come from agriculture. They come from pesticides, both the ones used in agriculture and the ones not used in agriculture. Some of them are industrial and mining products. Some of them are personal care products. Some of them are in waste products. And I put a display out of just some of the estrogenic ones over here that you can take a look at when you get an opportunity. If we take any of those categories, let's say the pharmaceutical ones, what kinds of pharmaceuticals are out there? Things like analgesics, painkillers of various kinds, antibiotics, anti-cancer drugs, anti-cholesterol drugs, Lipitor, things like that, antidepressants, birth control pills. You can guess this list can go really long. I just started at the beginning of the alphabet. From these various sources then of these different kinds of chemicals, some of them we ingest and they go into our bodies through our gastrointestinal system. Some of these we inhale and we absorb them through our lungs. Some of them we just touch them. We absorb them right through our skin, or we rub them on our bodies and absorb them that way. Once they get in the body, of course, they get distributed to everything that's in the body, and they end up in all kinds of secretions such as saliva, sweat, tears, semen. They end up in our urine and feces, and they even end up sometimes in our exhaled air from our lungs. Whoops. Again, when we talk about these endocrine disrupting chemicals operating as mimics or as blockers, they can work at all of these sites in the life history of a particular hormone. So the complexity, different endocrine disrupting chemicals may hit different parts of the system. Others may hit more than one spot or one part of the system. So they can show up almost anywhere. If we take and talk just about estrogens, and I'm going to pretty much talk about estrogens most of the rest of the morning. Estrogens have important roles in both males and females. They're important in brain development, they're important in the gonad development, they're important in other sex accessory structures, oviduct, uterus, so forth. They're also important in reproductive behaviors in both males and females. One of the things about development, and this scheme represents the development of the body during pregnancy from day 0 or week 0 all the way to 40 weeks, which would be normal birth. And what this shows is, among other things, it shows the development of our nervous system. And early on, the nervous system is developing, and somewhere between the 8th and the 10th week, the testis starts making testosterone, the male androgen, major male androgen. And that goes to the brain, and the brain converts it to an estrogen. And it binds to an estrogen receptor, and it makes the male brain work differently with respect to reproduction than the female brain. If this doesn't happen, the male brain will function like the female brain with respect to reproduction. If a female is exposed to estrogen at the same time in her development, it will also convert that part of her brain to the male pattern, and she'll be infertile because she won't be able to ovulate. If she's exposed to testosterone at that time, her brain will convert that testosterone into an estrogen and masculinize that female's brain. Just that narrow window in development. That's where I'm talking about timing is everything. If that fetus is exposed at this time, if a female fetus is exposed to an estrogen at that time, or this particular androgen, she just might get masculinized and be infertile. Now, estrogen excesses can have effects too, early in development. For example, too much estrogen can cause, in embryos and larvae and fetuses, can cause what we call sex reversal. It can convert an actual male animal to a female animal. Or oftentimes we get what's called intersex, where we have male and female tissues in the same individual. Probably just only hit part of the window, and we didn't get complete sex reversal, or it wasn't quite enough of the estrogen, and so we only got a partial sex reversal happening in that particular animal. Estrogen excesses in adults can have effects too. Contraception works in both males and females if they're exposed to estrogen, shuts down their reproductive system through feedback, and in males can actually cause feminization in adults. Now, the difference between what happens in embryos, larvae, and fetuses versus what happens in adults is that in adults it's reversible. We take away the birth control pill, and that woman usually will start ovulating again. Might take her a couple of times to get going good, but because she's been repressed for who knows how long. But the system responds back. These effects that happen with fetal exposure or larval exposure or embryos are permanent. We can't change them later. And it doesn't matter whether we're talking about effects of thyroid hormones or we're talking about effects of reproductive hormones. If they occur during fetal development, we can't change them later on. They're permanent. So I want to say a little bit about effects on wildlife, particularly focusing on some of our work here in Colorado. And you'll see how your bass from the Potomac come in here, because they're calling those intersex. I disagree with them, but that's neither here nor there. Vicki Blazer, who did that work, I've worked with her too. We don't quite agree on what we're calling intersex there. But it's the same kind of thing. Okay. When those reports came out, we thought we'd look at some of the rivers here in Colorado. So we picked 3 to start with. We've done more than that since then. But we looked at below Denver, city of Denver, below their wastewater treatment plant, and above, upstream. We looked in Boulder, at Boulder Creek, and we looked down in Colorado Springs. So we've got 3 different size populations, about 100,000, 500,000, and about 400,000 people, all sending their waste products to the same wastewater treatment, or to a single wastewater treatment plant for processing. Here's Boulder Creek, flowing along here, just immediately east of 75th Street. This is the wastewater effluent adding to the river at that point. Nice picture. We looked at white sucker populations living upstream and downstream of the wastewater treatment plant, which is located there. Of course, as you know, upstream, it's a gold medal trout stream. Downstream, it's 50 to 70% wastewater effluent. It's what we call a wastewater-dominated stream. In fact, almost all of our rivers in the western United States and a lot of the eastern ones are dominated by wastewater. If it weren't for wastewater, these rivers would not flow. What do we call wastewater effluent coming from a wastewater treatment plant after we dump it in the river? Any guess? How about river water? That's somebody else's drinking water downstream. Once it's processed, if it meets EPA standards, it then goes directly into the stream. What did we see? Well, upstream we saw a sex ratio roughly 1:1, which is typical for this species. Downstream, the sex ratio was 5 females for every male. Furthermore, both the males and females downstream were making fewer eggs and fewer sperm than the ones upstream. So we were seeing a contraceptive-like effect. We also saw that the males downstream and the juvenile fish, both males and females downstream, were making a particular protein called vitellogenin, which I've just abbreviated here. It's written out on one of your other slides. You'll see it. Vitellogenin is an egg precursor protein. It's made only by females under the influence of estrogen. Estrogen stimulates the liver to make this protein, and then it's incorporated into the eggs to make the yolk for the eggs. Here we find it in males and juveniles, but only downstream, not upstream. Furthermore, 20% of the downstream fish were intersex, and by intersex in this case I mean there was clear sperm-producing tissue and egg-producing tissue in the same individual, and both were developing. Very different from the picture of the bass, largemouth bass in eastern United States, where they just found isolated oocytes in the testes of the males, and that when they looked at older fish, there were none. So it was only in the younger fish that they saw that. But these are really intersex fish, and many of them were 50% female and 50% male in terms of their gonads. When we looked at the chemistry of the water, we found many estrogenic endocrine disrupting chemicals in the water, and I'll illustrate those in a little bit. When we looked at the water upstream, we couldn't detect them at all. And we were able to detect, for example, the natural female estrogen, such as estradiol, down to 1/2 of a nanogram per liter, which is pretty dilute. But there was a lot more than that, as you'll see. So one of the questions we got, is this new, or has this always been that way in Boulder Creek and the South Platte River and Fountain Creek down in Colorado Springs? Okay. By the way, the South Platte is 95% wastewater effluent almost all year long. That's Aurora's primary water supply. So I went to the CU Museum and I examined white suckers that have been collected in Boulder Creek 50 years ago and 100 years ago, and I looked at their gonads, and we could find no evidence of gonadal intersex. So we We're pretty sure this is a fairly new phenomenon, at least within the last 50 years, probably related to the increase in density of the population and all that stuff going through the same wastewater treatment plant, which hadn't been upgraded since about 1930. Point out, these are only correlative data, just like the data on the bass that they've collected around the country. It doesn't say cause-effect. It's just a correlation. And the reason we can't say cause-effect is there's too many differences in the upstream sites versus the downstream sites. Temperature's not the same. Dissolved oxygen's not the same. The fish are eating different stuff. There's other chemicals seeping into the river as you go downstream. There's no way that you can go out and compare two sites and say that the difference between the two sites is due to the fact that we added wastewater effluent to the one site. It's just a correlation. It might be a nonsense correlation. It might be a meaningful correlation. We have both kinds out there. I'll show you some other nonsense ones later. We went to the city of Boulder and said, can we go to your wastewater treatment plant and set up our little trailer and run an experiment to see how estrogenic your water is? And they said yes. The city of Boulder, unlike some places that I could mention, but I won't, says their attitude is if there's something wrong with our water, we want to know about it. We don't want to just put our head in the sand. So that's us right there. This is Goliath, and that's me down there. So what this allowed us to do is to sample the effluent going from the plant into the creek. And so we could pump some of that effluent into a holding tank above our trailer, and then we could pump clean water from upstream into another tank, and then we heated the river water up to the same temperature as the wastewater effluent because it's always much warmer. That's why the stream is so different downstream of the wastewater treatment plant, because the temperature is much higher. We don't usually find trout down there, for example, at all. They don't do well in warm water. So we heat the water up, then we gravity feed it into our trailer, and remind you that the effluent is 100%, and the river water downstream is roughly 50% effluent. And so we filter it, and we can mix it in whatever proportion we want, and we can run it down through these various aquaria. For example, these aquaria are getting 100% creek water or reference water. These are getting 50% mixture of the two, and this is pure effluent coming down here. Notice it's a little cloudier too. And then we studied, instead of white suckers, since white suckers are pretty big, we picked what we call the EPA poster fish. It's being used for this kind of research all over the country, in fact all over the world. But these fish are actually native here to Colorado. And we took breeding male fathead minnows that were in the peak of breeding condition. And we put them in these aquaria to see what would happen when they were exposed to 50% or 100% wastewater effluent. So what happened? We measured a whole bunch of stuff, and I'm just going to give you a couple things. If you want to have a more detailed analysis, you could pick up one of these down here afterwards. But this is one characteristic. It's called a nuptial tubercle. It's a male sex character. It's actually used in reproduction. There are little bumps on the snout of the male. They are androgen dependent, and they regress in the presence of estrogens. So we looked at those, counted the number of them, as illustrated here, how many were on the fish, and we saw that they dropped off rather dramatically in both 50% and 100% effluent, and it was statistically significant within 7 days of exposure. Here's the production of that vitellogenin protein I mentioned before, and we found that in this case, in the 50% effluent and the 100% effluent, the fish were making huge amounts of this protein, equal to what a female can make when she's actually making eggs, which is indicating this is the maximum stimulation. Doesn't matter how much estrogen we give them. We'd never get any more than that without killing the fish. And that was statistically significant. Even probably before 7 days, we didn't look at 4 days, which we could have. Now, what were the estrogens that we measured in the effluent? You heard about nonylphenols. Nonylphenols are breakdown products from detergents that are actually created in the wastewater treatment plant process. They're also found in all of your spray cans, the inert ingredient that's used to disperse whatever's in your spray can, particularly if it's a lipid, are usually nonylphenols. We have estrogenic pharmaceuticals such as diethylstilbestrol. I'll say more about that later. Ethinyl estradiol from birth control pills. We have chemicals called phthalates. At low concentrations, they're anti-androgenic. At high concentrations, they're estrogenic. Because if you're a male, it doesn't matter whether you're hit with an anti-androgen or an estrogen. They tend to both reduce your masculinity either way. Phthalates are found in lots of things, soft plastics particularly. The standard water bottle that you buy your water in, the small bottles, they tend to leach out from those. They're found in children's toys, baby toys particularly. They're found on the dashboard of your car. If you wonder what that awful film is that you constantly have to clean off the inside of your windshield, and you say, this doesn't make any sense. Nobody in my family smokes, and what is this crud that keeps ending up? It's the phthalates coming out of your dashboard when the sun warms up your dashboard, and what you didn't inhale is now stuck to your the glass. And they're found in a lot of PVC piping, particularly the medical tubing and medical bags tend to leach out these phthalates into whatever you put in them. There are also the natural estrogens that every one of us in this room excrete. A male excretes about 8 micrograms a day. A female who's reproductively active produces about 30 micrograms of estrogens a day that she excretes. A postmenopausal woman produces about 8 micrograms a day, same as a male. And a pregnant female produces about 2 milligrams a day. 1,000-fold more than a non-pregnant female. Or a woman who's on birth control pill excretes about 30 micrograms a day of the birth control estrogen. 1,000-fold less than she would be excreting if she were pregnant. I ended up on the Catholic Church website as a advocate for stopping the use of birth control pills because they were in our effluent and they were feminizing fish and wildlife. But the point is that if those females were pregnant, merciful heavens, they'd be producing a lot more estrogen. And you know, that was not the point I wanted to make at all. They took me off, finally. There are plant estrogens. A lot of plants make estrogenic chemicals. And we ingest those plants, and particularly soy products contain a lot of this one, genistein, which is one of the more common ones. There are a number of estrogenic pesticides still out on the market. The one that we replaced DDT with methoxychlor, is also estrogenic. Not as estrogenic as the DDT was, but still is a problem. And then this one, bisphenol A. And I'll tell you a little bit more about bisphenol A because it's getting into the news finally a little bit. Where do we find it? It's in dental sealants that we put in kids' teeth. The polycarbonate plastics, you might see that some of them are now labeled BPA free. Guess what? They still leach estrogenic chemicals, just don't leach BPA. Thermal inks, cashier receipts that you get. You absorb the ink, you absorb the bisphenol A right from the ink when you handle them. It transfers to your money. And dollar bills are all contaminated with this BPA. They're in the resins that line all our food and beverage cans. They're in CDs and DVDs. They're in PVC pipes, particularly the really hard ones. And, oh, that's currency. I already said that. They're all over the place. We produce over 1 billion pounds a year of just this one chemical in the United States alone. And we're seeing a lot of headlines linking BPA to various reproductive disorders. Correlations, correlations, correlations. Are they nonsense correlations or are they meaningful correlations? And we'll address that a little bit later. The really important take-home message from all of this mixture of estrogens is that all of these particular compounds are capable of binding to the estrogen receptor, which means that they're additive in terms of their effects. So if you have 5 chemicals, each one at 20% of the no effect level, that the toxicologist would tell you, 20% times 5 adds up to 100%. You get the effect, even though you can't blame any one of the 5 compounds, because it's the mixture that produces the effect. And estrogens are a real problem from this point of view. Not all of the endocrine disrupting chemicals work quite that way, but the estrogenic ones, most of them work through the same mechanism, binding to that estrogen receptor. So any that you have adds to any others that you have. So it's the total exposure, not what you're getting from one source, that is important. Now, in Boulder Creek, what we found in that first experiment, and we repeated it the next year, in the first year that we did the experiment, we found that we could, since we knew what the potency was of each of these different chemicals to bind to that receptor, we could then take that information, know the amount that was there, and we could then say how much of it is equivalent to the natural estrogen that we normally produce. So we could do what we call an estradiol, which is our major female estrogen, estradiol equivalency. And so when we did that, it was equivalent to about 50 nanograms per liter of effluent. How much effluent is coming into Boulder Creek? About 17 million gallons a day. 100,000 people get up in the morning and flush the toilet. That's a gallon a shot. There's 100,000 gallons of 6 to 12 hours accumulation of estrogenic chemicals all hitting the wastewater treatment plant at the same time. A facility that was not designed to deal with chemicals at these concentrations, even 50 nanograms. Now, okay, obviously 50 nanograms feminized the fish in our, and demasculinized the fish in our system at the wastewater treatment plant. What does this mean for out in nature? Well, Karen Kidd did a study up in Canada. We couldn't do this study here, never get it approved, but they got it approved up in Canada, and they actually treated a whole lake with ethinyl estradiol from the birth control pills, EE2 we call it, at a concentration of 6 nanograms per liter. And they treated that lake for 3 years and compared it to a control lake and looked at a number of species of fish, but I'll just tell you about the fathead minnow. Here are the results. Here's where they started treating this year, and this is what happened to the population. They wiped it out in 3 years through primarily contraception effects in the females and lethality in the males because they accumulated all this vitellogenin. They didn't have any place to put it, clogged up their kidneys, and the males died out first, really. And then the females didn't have any males to mate with, so that made it double hard for them. And they couldn't make any eggs anyway, so it didn't really matter. They were gone. Now they're following the reintroduction of the fish coming back into the lake and to see how long it's going to take it to recover. But of course, in rivers and streams, we're dumping that stuff in constantly every day.